Another high-yield topic on the board exam is that of DNA repair. It's very likely you would get a question which asks you to understand these mechanisms of DNA repair. So we have three of them that are the highest yield for you to be asked about. They are thymine dimer repair, mismatch base repair, and cytosine deamination. Thymine dimer repair is usually caused by UV radiation. So every time you step out into the bright sun, after you've been studying biochemistry all day, you could think about how your DNA is going to be repaired because you have enzymes to do so. Unfortunately, some people inherit a defect in the enzyme responsible for this repair. The enzyme responsible is called excision endonuclease. And if you inherit a defective copy of that gene, well, you could end up with xeroderma pigmentosum, a very high yield disease. Some of the repair enzymes are DNA polymerase and DNA ligase that are going to come in after the lesion is recognized by this excision endonuclease. I'll show you some illustrations that highlight this. Mismatch base repair is another type of repair mechanism that happens, whereas thymine dimer repair tended to happen during the G1 phase. Mismatch base repair is going to happen during the G2 phase, after the DNA has already been replicated through the synthesis phase. So mismatch base repair is very important. DNA polymerase by itself, with excision endonuclease repair, still makes a mistake on the order of 1 in a million. That's pretty good, 1 in a million. But it's not good enough because remember, we have 3 billion base pairs in our genome. So in order to get that added level of mutation prevention, the cells have a mismatch base repair system which goes back after the DNA has already been replicated. It makes sure that the bases are paired properly. So in mismatch base repair, there's at least 2 genes. One of these is called HMSH2, that stands for human MSH2, and HMLH1, human MLH1. These initiate a repair mechanism wherever mismatches are recognized. After these mutations are recognized and corrected, then DNA polymerase and ligase can come in and fill in the gaps appropriately. The clinical connection here is at the bottom, and it's called hereditary non-polyposis colorectal cancer, or HNPCC for short. Hereditary non-polyposis colorectal cancer. There's another name for this, and it's called Lynch syndrome. So in this particular malady, individuals have a defect in the repair of their mismatched repair after synthesis. So in HNPCC, what you see is accumulation of mutations, i.e. more solid tissue cancers. So somebody with Lynch syndrome is going to be predisposed to early cancer. Another repair mechanism which is tested is cytosine deamination. Going back to the structure of DNA and nitrogen bases, what did we see when cytosine became deaminated? Well, what we saw when it became deaminated was uracil. Remember, the loss of that amino group converted this into uracil. How does this happen? Spontaneously. Spontaneously, during the course of a day, many of the cells within your body will spontaneously get deaminated at their cytosines, thus creating uracil. Thankfully, we have a uracil glycosylase and an AP, which stands for apyrimidinic endonuclease, which can come in. It will cleave out the sugar that this uracil is bound to, and the AP endonuclease will cleave the phosphodiester bond, and then eventually the polymerase and ligase will fill in where the cytosine deamination step had occurred. So the net effect is that you don't want U's in your DNA. You only want C's and T's. So if the cytosine gets deaminated, it becomes uracil. Uracil has no business being in DNA. So through the action of uracil glycosylase, which will cleave the sugar-nitrogen base bond, and then AP endonuclease, which will cleave the phosphodiester bond, you remove that uracil, and you can lay down the cytosine back where it should have been. It would be nice if there was a an amination step that could happen, but it doesn't work that way. We have to chop out the base and lay down new cytosine where that base once was. So here's your high yield picture of thymine dimer repair. So you have thymines that are adjacent to one another, not across from one another, they're adjacent to one another. And there's an enzyme that's called excision endonuclease. Notice that these two thymines have formed a covalent bond. That should not happen. Thymines that form adjacent to one another should remain separate from one another. But if UV energy comes in, it could shuffle electrons around. So now that there's a covalent bond between these adjacent thymines, that is a problem. The next time this cell becomes replicated, the resultant DNA is either going to be mutated or it's going to be truncated in some way. This is a major lesion on the DNA sequence. So the excision endonuclease is going to come along. It's going to recognize this lesion, okay, and it's going to clip out. It's going to clip out the DNA here. Other pyrimidines can form dimers. In other words, you can get cytosine-cytosine dimers. In double-stranded RNA, you might get uracil-uracil dimers. That's not clinically relevant. For our purposes, it's thymine dimers are the only pyrimidine dimers you have to worry about. So if you're given a question that says pyrimidine dimers are forming, you should automatically assume that it's thymine dimers they're referring to. Now, the disease xeroderma pigmentosum. The disease xeroderma pigmentosum occurs when an individual inherits defective copies of excision endonuclease. This is an autosomal recessive disease, so you need not just one mutant, but 2 mutant copies of that enzyme in order to express the disease, the phenotype of the disease. So, how would you recognize one of these from a case on test day? It's going to be a patient that's very pale. Why? Because they're sensitive to UV light. They have a lot of freckles because every time they do get exposed to UV light, they're going to get mutations going on. They have multiple skin cancers, and they're even going to get corneal ulcerations because they don't have the ability to correct mutations that happen when they're exposed to UV light. These folks are going to sort of live a vampire existence because they want to come out only at night. So as illustrated here in the figure, you see that the net effect of the repair mechanism is that the removal of the covalent bond between the two thymines is accomplished and that the DNA gets laid back down in its place. That's shown here. You see that the affected mutation, the thymine dimer here, is clipped out by the excision endonuclease. Along comes DNA polymerase, and what's it going to do? Just like every polymerase here, it's going to read the template from 3 to 5. It's going to synthesize the DNA from 5 to 3. And the last little phosphodiester bond, that last little nick, how does it get filled in? It gets filled in with DNA ligase. You're going to see DNA ligase repeat itself over and over throughout this reviewed series. And the role of DNA ligase is always the same. It's to reform the very last phosphodiester bond to seal in some sort of nick that was created in the DNA. Now we need to discuss some additional genes that are important for maintaining the fidelity of DNA replication. After all, the name of the game is making sure that the DNA that gets copied is identical to the DNA you started with. So, the various mechanisms we already discussed include mismatch repair. This happens in the G2 phase, and if you remember, there were 2 genes, MSH2 and MLH1, which are responsible for ensuring that any mismatch bases made during the S phase are corrected. And when one of these genes was defective, it led to HNPCC, which is hereditary non-polyposis colorectal cancer, or Lynch syndrome. The name is a little bit misleading because you will see colon polyps in HNPCC, but the name is given to differentiate it from another disease called familial adenomatous polyposis, or FAP. So you will see some colon polyps here, but not as many as you see in the other disease, which is familial adenomatous polyposis. You will review that topic in the pathology section. All right, the other repair mechanisms include thymine-dimer repair. Remember that the covalent bond between the two thymines, if it happens during G0 or G1 typically, has to be corrected. If that excision endonuclease is missing, the patient is going to have xeroderma pigmentosum, autosomal recessive disease. If cytosine deamination is not present, then the patient's going to accumulate uracil residues, and if they don't have the uracil glycosylase and the AP endonuclease necessary for that event, well, then they're going to accumulate mutation. What we need to add in here are some additional genes that control entry into the S phase. You see, what the cell needs to do is to block somehow the accumulations that accumulate during the G1 phase. Cells will accumulate these mutations, and the mutations will be repaired, but it takes some time. And sometimes the cell still has a lot of mutations occurring and is not ready to jump into the S phase. So, these mutations have to be removed before the cell enters the S phase, because once the cell enters the S phase, you're going to duplicate the mutations. They're already going to be there. They're going to be present in every cell derived from that parent cell. So, two genes we're going to focus on are P53 and retinoblastoma. P53 in particular being the most important. So P53 and retinoblastoma are examples of tumor suppressor genes. Let's define what a tumor suppressor gene is. A tumor suppressor gene, when they were initially identified, were genes that, when they were mutated, the cell would accumulate tumors. If the protein was put back into the cells, it would suppress the growth of tumors. Well, it turns out that the actual functions of these tumor suppressor genes is to encode proteins that slow down the cell cycle. And they slow it down so that it allows time for the repair mechanism to try and do its job before the DNA becomes duplicated in the S phase. So P53, think of it as it blocks. It stands there like a barricade, and it blocks the entry from the G1 into the S phase. Retinoblastoma works fairly similarly to that, but P53 being the most important one. And actually, having said that, there's another function associated with P53 besides its role in slowing down the cell cycle and initiating DNA repair. And that other mechanism is that P53 can actually induce apoptosis. I think of P53 this way. It's sort of like a mob boss, a crime racketeer boss. And it's sitting here, and it's going to slow down the cell cycle. It's going to slow down the cell cycle, but it's also going to send out some troops. It's going to send out a crew that just in case the job doesn't get done correctly, just in case the cell cycle doesn't get slowed down and things get repaired the way they should, it's going to whack the cell. The cell is going to get whacked. What do I mean by that? Well, P53 is going to induce apoptosis. So if the cell cycle doesn't slow down, if the DNA doesn't get repaired, what's going to happen? P53 is going to signal the cell to kill itself. So that's how P53 works. There's actually going to be viruses that you'll see later on that have an ability to make proteins that interfere with P53 function, that interfere with retinoblastoma function, and that's what actually causes some of these viruses to cause cancer, to be tumorigenic. In particular, the hepatitis B virus, in particular, human papillomavirus. They have proteins which can sequester either P53 or retinoblastoma, and that allows these cells to cause cancers. A topic that we have not talked about yet is microsatellite instability. So let's define what a microsatellite is. A microsatellite is simply di and trinucleotide repeats that occur throughout the genome of an organism. Typically, it's in areas of the genome that are non-coding. And what I mean by that is it's areas of DNA that don't contain information that will be transcribed and translated into protein sequence. The relevance of microsatellite instability is that cells that lack mismatch base repair, because either they've acquired mutation, a person's inherited mutation defects in those genes, or they've somehow acquired a mutation through the environment to one of those mismatch base repair genes. Well, in those individuals, the number of microsatellites will remain the same, but they'll have microsatellite instability. And what microsatellite instability means is that the number of di and trinucleotide repeats within those specific regions will tend to be different from one cell to another. In a healthy individual, you should have the same number of microsatellite repeats within a particular area of your genome in all your cells. But if you tend to have a defective mismatch base repair system, the number of microsatellite repeats in a given section of your genome is going to be different among each of your cells. So that's how they could ask you about that. They could describe in words or in a figure a specific region of DNA and tell you that the number of di and trinucleotide repeats is different in this region from one cell to another. They might ask you something about Lynch syndrome or HNPCC. They might ask you about the name of the gene for mismatch base repair, remembering that it's HMLH1 or HMSH2. So remember microsatellite instability. And as we talked about earlier, Lynch syndrome, which is another name for hereditary non-polyposis colorectal cancer, is caused by a defect in one of these mismatch base repair genes, HMLH1 or HMSH2. And in these particular cells, you're going to see microsatellite instability. That's the major tie-in here. That's going to conclude Chapter 2 for us. So we talked about how DNA replicates itself, the various modes of prokaryotic versus eukaryotic replication, and very importantly, we discussed the repair mechanisms involved. So we have thymine dimer repair, cytosine deamination repair, and also we have DNA mismatch repair. Additionally, two very important proteins to consider for this replication repair process are the tumor suppressors p53 and retinoblastoma, because these are going to help slow down the cell cycle and allow all these repair mechanisms to carry out their function.